So let's look into the another method of this multiple sequence alignment. We call it as a progressive method because things they progress one after another in a progressive fashion or a progressive manner. So that's why it's called as a progressive method, which has been implemented in most of these multiple sequence alignment softwares. So it's a heuristic method. Again, I would repeat what is a heuristic method. So that gives you a quick and efficient solution to your problem. It may not be exact solution, but serves your purpose. So at first we align two closest sequences and then add next most closely related sequences until we get all the sequences they are aligned. So we compute those pairwise distances scores for all pairs of sequences. So it's kind of you are using what we have learned in a previous section, this sum of pairs scoring schema. We generate a guide tree, and this tree ensures that similar sequences are closer on the tree. So what you do next is after you get this tree, you go back to your sequences, and now you use this tree as a guide to guide your alignments. So here is how it works. We have pairwise alignments of all sequences. You can do with any method. Local, if you are aligning some sections or segments within your complete sequences. It can be global, where you can take the whole sequences and align them one another depending upon what is your question. Are you planning to do this multiple sequence alignment locally or globally? You're looking for motifs or patterns, then your possible scenario where you will be using this local alignments. If you're doing with your global kind of problems where you are aligning the sequences whole genes with one another, so in that case you would do this global, and you know the algorithms behind them. We can go with dynamic programming approaches where we can take the whole problem, break it into sub problems, and then alternatively you can go with heuristic methods. So with any methods you can come up with pairwise alignments. So we get these pairs. For example, in this slide there is a distance matrix. So distance is calculated as 1 minus number of identical residues divided by number of overall aligned residues, or what total residues are present in this alignment. So here we have on x-axis and y-axis we have the same sequences. So we have in this example, which we took from this reference, they use those hemoglobins and related proteins, myoglobins, etc. In humans and other related species. So we have on x-axis same sequences, on y-axis we also have the same sequences. We don't want diagonals, since diagonals are the places where the sequences are comparing to themselves. So that's actually we don't want, because there is no distance. And same way we take the half of the matrix, because this remaining half is actually the duplicate version of the same what we have on the other half. So for example, if you take sequence number 1 and compare it with sequence number 2, so they are pretty close and their distance is less, it's 0.17. Same way we can fill up the whole matrix with one another. So these distances are then obviously we have come up with a matrix where we can have the distance between two species, and we use this information to construct a phylogenetic tree, which is right here. And we will go over the phylogenetic tree in next lecture, which is coming a few slides from now. Just for this, since our broader topic here was progressive alignment, so you just look into, from those distances we can come up a tree where we can see that HBB with human and HBB with horse, they are pretty close with one another. So on the tree we have these different sections, we call them clades. So actually HBB human and HBB horse, and they are in this clade, they are with one another, and in this way the rest of them they are coming one after other. So probably this the LGB2 lupla is having the highest distance from rest of them. So more close to species are, they are more close on the tree. So when we start aligning them, we will probably start with these number 1 and then number 2, and then we go for 3, then we add the 4th, 5th and 6th. So in this way, now we align those sequences, we came up with their tree. Now we are having this guided alignment, so that is guided by the tree. So while looking into the tree, we will start aligning these sequences again and again. So we'll take the first two, which was in the previous tree, like number 1 and 2, we put them together, then we add the third, and so on. We keep adding the rest of them unless these alignments are complete. So this method is progressive method. We progress one after another step. So this method is actually a fast, efficient, and the alignments are reasonable. In many cases they are acceptable, but the disadvantage is not the exact or the optimal solutions, heuristic solutions, which are quick and fast solutions, but which may not be exact solutions, may not be that much true. So accuracy is sometimes your concern if you are using these methods. Sometimes there might be some errors which are incorporated by different steps you follow in this algorithm. So overall, a progressive alignment method is a heuristic method. We can progressively align the sequences, we align them together, we come up with a tree, and we use that tree as a guide to do the multiple sequence alignments.